It's 9 o'clock, and we are ready to kick off day 2 of the 2023 BioStock Life Science Summit event. A warm welcome to all of you here, early risers. Very nice to see you. Some of you were here yesterday, and some of you weren't. But I can assure you we have a jam-packed program for today. Lots of interesting presentations and a few equally interesting panel discussions. And we will also finish off with a live interview with WntResearch, which is one of our proud sponsors of the event. I forgot yesterday to introduce myself. I'm Jonas Söderström, and I'm the CEO of BioStock. So on behalf of myself and the fantastic team that has been working really hard to put this event together, I'd like to thank you for coming, and also just leave the word to my brilliant colleagues and moderators, Cecilia and Michael.
Thank you.
You can start the show.
Thank you. Yeah, let's just do some information before we start with the first speaker. Just like yesterday, the event is filmed and photographed, just so you're aware of that. And we will be doing 10-minute presentations followed by a 5-minute Q&A. So with that, I think we are ready for the first presenter of the day. And I'm happy to introduce Peter Nahlstedt, the CEO of SenzaGen. Welcome, Peter.
Well, good morning, everyone. Nice to speak with such a almost full big room. I'm Peter Nahlstedt, CEO of SenzaGen, and we are a sustainable growth company in a market undergoing a paradigm shift. We have high-tech solutions that help companies transition to non-animal testing. Thereby, we help companies bring safe, ethical, and more sustainable products to market. We also help companies secure a more safe production environment for their employees, and we help the world to reduce the number of animal tests. And we are on a growth track and have very strong execution in the last couple of years. This year, we have rolling 12-month sales growth of 27% plus. The first 9 months of this year, we have 16% plus growth, and we are quickly approaching break even. Our gross margin, we were able to expand whilst driving revenue to 69%, that's plus 5 points. And we also improved our EBITDA margin to negative 29%, which is a big improvement with 17 points. So as you can see, the company is moving towards break even. We received approval for our own developed, springing from Lund University immune technology called GuardSkin from OECD last year. And we have made 2 acquisitions of growing and profitable companies to complement Guard. And we have also in this year recorded the first revenue and innovation synergies. We also received record high numbers for the Guard platform from major cosmetics, and major chemical industry, and big pharma companies throughout the world. And as you can see on the right-hand side, this is translating into quick growth. We are now a group of 3 companies at SenzaGen. We have SenzaGen in Lund, with expertise in skin sensitization and toxicity testing. Developer of a state-of-the-art Guard technology, 20 employees, 8 of them PhDs, based here in Medicon Village. We have a subsidiary called VitroScreen, which is an expert CRO in preclinical efficacy and toxicity testing, experts in advanced human 3D tissue models, 10 employees. And we have also complemented our operation with a consulting and advisory firm called ToxHub, which possess toxicology and pharmacology regulatory expertise. And the reason for this acquisition is, of course, that customers need advice which tests to run. When they have run the test, they need help with compiling regulatory dossiers, and that is sort of bringing a more complete solutions for our clients. We work on a global market, the in vitro toxicology testing market. It is growing in the last 5 years with 7%. We see signs that it will continue to grow. The total market size was estimated at $9 billion. We think it will grow a little faster in the next period. It's driven by more animal testing bans in more countries, ESG engagement with large companies, human relevance, and cost and time. The biggest markets are Europe and North America. That's where we focus. And as you can see, we are pretty industry agnostic. We work with cosmetics, medical device, chemicals, pharmaceuticals, but also nutrition and food additives. We have a simple growth strategy. We work with high technology, best-in-class in vitro technology, which we are aiming to replace animal testing with. We establish those technologies as standards, and thereby we bring safer products to society. And we have a split strategy with organic growth, where we focus on developing sales channels and good and deep customer relationships, and acquisition growth, where we focus on finding complementary technology and complementary value chain offers like ToxHub. But it started with the GARD technology, and I want to take you through this slide about GARD. It's important to get a grasp of what we're doing. We work with a biological cell system, in this case a human dendritic cell line. This cell line has been exposed to a training data set of known skin allergens. Thereby we have developed a biomarker signature, which has been locked, and this biomarker signature and was trained with skin or machine learning. And then we have arrived at the prediction model, which is an algorithm, and this algorithm is of course locked. It's put into an app, and this is our testing platform together with our proprietary cell system. We perform this test here in Lund, but we also perform it at a couple of different CROs where we have performed a license deal. And the GARD technology has big advantages over what we call first generation in vitro testing, but also, of course, big advantages over in vivo testing. So the GARD technology is based on 196 biomarkers. It gives a superior accuracy of between 90 to 95%. It can be used for a very broad applicable set of substances, whilst comparative to traditional in vivo testing, the relevance to human data is about 70 to 75%, not very ethical and also very time consuming. So there are big drivers to move towards more advanced technology, and GARD is the most and only genomics-based tool that is available on the market. This is getting success with clients. We work with large multinational clients. Essity in Gothenburg is one example of a client. They are very happy with GARD working for their absorbent hygiene products. And we also have another medical device client called Sonova. It's a Swiss hearing aid company, one of the largest in the world. They also use our test to check for new materials when they are implementing that in production. And as you can see on the right-hand side, or well, what is it, left-hand side for you, we have a number of collaborations with very large clients like ExxonMobil, Corteva, Lundbeck in Denmark, and IFF in the nutrition space. And just giving you a view of our business model, as this is not a market you might know so much about, we know that it's a pretty long sales pipe. We start with contact and discussions. It usually takes between 6 to 18 months until we arrive at the first order. First order is in around 300,000 or slightly shy of that, Swedish krona. And then we know that the repeat purchase rate year 2 is between 30 to 50%, and then the purchase sum on average is about 510,000 kronor. And our model is basically very simple. We want to add more customers on this platform. And we have been able to do so in this first 9 months. We added 25 new customers in 2023 versus 18 for the full year 2022. This is, of course, resulting in sales growth, 23% with GARD this year. But we also have 77% of our revenue from recurring clients. So as long as we can keep on doing this, the company will grow. And we did get new clients and some new key orders in this first 9 months. First of all, we have a repeat order from the Research Institute for Fragrance Materials in US, RIFM. It's worth 1.6 million. It's a development of GARD where we can also check for photosensitization. This means that a substance can change its it can become allergenic or dangerous if it's exposed to UV or sunlight. And our GARD platform can now, what we're looking at is if we can see a difference between photoallergens and photoirritants. It's a big deal in cosmetics and a big deal for these chemical companies that develop products for that space. So not only is this a valuable order for us because we just received it, it's good for our sales this year, but it is also a shopping window to the 40 or so members of the RIFIM organization, which is the major chemical companies in US and Europe. We also had a large follow-on order from the world leader in cosmetics. It's an adaptation project where we make a specific version of GARD to fit their needs. And then we released this morning, we got a new order from a world-leading client. We cannot name them, unfortunately, but they are in the fast-moving consumer goods sector. The order is worth 800,000, but it's very strategically important because, as I showed you on the previous slide, we have a high repetition rate, and these large multinationals is the type of client we want in our portfolio. And we have in our daughter company in Italy, an increased demand for efficacy testing in pharma and cosmetics. So the first half was good. We had 18% growth and an EBITDA loss which was reduced. I also want to point out that we had a cash position at the middle of the year of 25 million. And with this increasing sales and having our costs under control, our burn rate is significantly reduced, and we think we have enough cash to take the company into break even. So what's the next steps for SenzaGen? Well, we want to continue our growth story, driven by the GARD platform. We also have a platform at VitroScreen called Aura. It's a 3D model platform for more advanced model testing, efficacy testing in pharma, and advisory services. We want to keep our costs under control, expand the test and service portfolio, and then execute on our R&D program to get more approvals and increase the market for GARD. And there are more opportunities for us, and the development is going our way. We have the FDA Modernization Act in US, where they removed the mandatory animal testing before proceeding into clinical trials. And we have the medical device regulation here in Europe, and the ECHA, which in the law text now says to the industry that if there is a non-animal alternative available, you should use that. And we have the best high-tech solutions for it. So thank you very much, and welcome to SenzaGen's growth journey.
Thank you, Peter. And we have quite a few questions here, and people want to talk about money and profitability, unsurprisingly. So, Peter, what needs to happen for SenzaGen to reach profitability?
Well, we just need to continue exactly what we've been doing for the last 2 years. Get more customers, keep our costs under control, and keep on maintaining gross margin. And I think it's possible. We are adding, like I said, 25 clients this year. Another one will release, I guess, 26.
So when you have reached profitability, what kind of margins are you aiming for?
I think SenzaGen we now reached a gross margin of almost 70%. I think that's a good goal to try to maintain over long term with our proprietary technology. But when it comes to profitability, I think, EBITDA margin of 20 to 25% is feasible.
And if we look at what could potentially hinder you from getting there, there's a question here about what you see as the main hurdles or the main challenges for SenzaGen to grow your market size and to break even.
I think, as many companies here, we work with brand new technology. And life science is cutting edge, but it's also conservative. And I think toxicologists in general are pretty conservative profession, which I think we all should be very grateful for. So it takes some time to build up the scientific evidence about the platform to get more and more people to adopt new technology. And that time frame is is maybe the biggest hurdle for us. But I think beyond that, we have the approval in place, we have the organization in place, so we should just keep on executing.
How do you work to convince the toxicology profession to use SenzaGen's product?
Well, we think the best salespeople are our customers. So what we do is that we try to create collaborations with the world-leading companies like Rifim or with Exxon Mobil, and we arrange joint presentations, joint scientific abstracts for the main conferences, where we have them to present the findings about GARD and not us. That I think is the number one. And then, of course, we work with very tight relationships with the large companies.
We also have a question about ToxHub here. They joined your group in December, I think it was. How would you evaluate the first couple of months?
Well, we started off really great. ToxHub had great sales in the first part of this year. We were very pleased, and it's also a very profitable operation. I think the EBITDA margin is close to 40%. So it was a great addition to our group. In the last 6 months, it's been a little challenging because the MDR, which I also think a lot of companies here are grateful for, was extended until 2027. That meant that their customers took it a bit more easy with some of the advisory. But we are building more business now, and we see that we can combine the GARD sales approach with also selling ToxHub services, where we have the first few orders in already.
So you're expecting that to become less challenging?
Yes.
Well, thank you so much, Peter.
Thank you very much. Thank you.
Right. Then we're happy to welcome up second speaker of the day, the CEO of Cantargia, Göran Forsberg. Welcome, Göran.
Thank you so much. Pleasure to be here in Lund and present Cantargia. And when you have an annual event like this, it's always nice to reflect, have we had a good year or not? And I have my personal view is that we had a very good year at Cantargia, so it's a pleasure to give you this update. So for those of you who don't know Cantargia, what we're completely focusing on is to develop antibody therapies, and we have a unique niche in that we discovered a novel target called IL-1RAP. And personally, I think the more I learn about IL-1RAP, the more I see the opportunities, and the more actually I love this molecule, because there is so much you can do here when it comes to unmet medical needs. So, for instance, it's overexpressed in cancers, and it's also a great opportunity here in autoimmune inflammatory diseases, and we're trying to make profit out of both. And in cancer, we treated almost 300 patients now. And we start to see clear signals of activity in several difficult-to-treat cancers, both pancreatic cancer, lung cancer, and now lately TNBC, which is triple-negative breast cancer. So it looks really, really promising. And we also have an ongoing randomized trial in triple-negative breast cancer, where we hope to have results end of 2024. And we have a phase 2b trial in preparation in pancreatic cancer, also a very important controlled trial. And in CAN10, we're in phase 1 clinical development, and the idea is to work towards systemic sclerosis and myocarditis later on, and results continuously doing this trial to make sure that the market is updated. We're also in a good financial position. We have 287 million Swedish on the bank right now. And even if we have ambitious programs in front of us, it will take us until end of 2024. And we also have very strong owners in several Swedish institutional investors backing us. So if you look at the pipeline, you can see that it's pancreatic cancer, triple-negative breast cancer on phase 2 development, and lung cancer. We recently completed recruitment and are going to present results once they mature, CAN10 in phase 1. But you can also see that we have emerging projects here. So once these late-stage assets evolve and get partnered, we will also be able to finance new opportunities here. So very interesting future for the company. And very quickly go through the clinical results. And in pancreatic cancer, we treat patients in first line. So unfortunately, most patients are diagnosed with a metastatic disease and a very poor prognosis, with an expected survival of, 8, 9, perhaps 10 months in different trials when you treat with chemotherapy. We treated more than 70 patients. We see much longer survival, 13.2 months. We can also see, if you look here, that lots of patients have very deep responses, which means that they have a very good tumor shrinkage, which also is during a very long time. So a very good result. But even more excitingly is that we can see that the patients that have the highest levels of our target are actually the ones driving the efficacy signal. So here we can see that patients with high IL-1RAP have more than 14 months overall survival. You can see that here on the waterfall that they have the really deep, nice responses compared to the ones with low IL-1RAP. And this is what you expect if you have some kind of robot which is getting into the tumor. Of course, the more target you have, the better the efficacy should be. What I don't have time to show is that if you don't treat these patients, you would expect the opposite. These patients with high IL-1RAP are the ones with the worst prognosis. So if you want to make it very simple, you can say that you made the losers to become the winners with our therapy. So a very strong result and something we are definitely building on in the upcoming clinical trial, which has now been designed according to new FDA guidelines, where you need to study 2 different dose levels in a randomized trial to have full documentation of that. So we're going to study the 2 most promising dose levels, which are 80 and 200 milligram, in combination with standard chemotherapy for first-line pancreatic cancer. And then the control group will be patients getting chemo only. And we will also look subgroup of patients for high IL-1RAP versus low IL-1RAP to get these results once and for all in stone. And I think that will, with that type of result, we will be in a very, very strong position. We also have strong results in lung cancer, and I don't have time to go through all this in detail, but I'd like to point out that one finding, so we have very good response rates, we have good survival, but one thing which is striking here is that we have several patients with complete responses. This is extremely rare in non-small cell lung cancer, and this is obviously a very strong signal. So what we're trying to figure out now is exactly how to find these patients, because it's a very competitive environment. And if we have good biomarkers to find these patients, we will obviously be in a very good position to have a discussion with potential pharma partners around this. And last week we presented new results in triple-negative breast cancer, and the story is very similar here. We get good results, we get very deep responses, much better than you would expect from chemotherapy alone when we do chemotherapy combination. The progression-free survival, which is how long time does it take for cancer to start growing again, is longer than you expect from chemotherapy alone. Survival it's still very early, most patients are still alive, but the signal is here. It really is a clear trend that we're prolonging survival. And here we are in a fortunate position that the phase 2 randomized is already ongoing and recruiting well. And hopefully recruitment can stay really good. So obviously that once we have results in this trial, we will go out and communicate the top-line results as quickly as possible. So to summarize very quickly, we have very exciting target. We have a very exciting mechanism of action here. And we see effects in 3 different cancer forms now that have, let's say, certain criteria in common. They are driven by specific biology, and they have lots of IL-1RAP. So we really start to carve out a niche here where we can deliver value to patients and hopefully to shareholders. So then our second project, very quickly, CAN10. It's also binding IL-1RAP, but it's doing it in a very unique way. So if we look at the nadunolimab, it's primarily blocking the IL-1 signal. Here we are actually blocking 3 different signals, IL-1, IL-33, and IL-36, which are all driving different diseases. So IL-33 is, for instance, involved in asthma. IL-36 in various skin diseases like psoriasis, and IL-1 is almost always involved in inflammatory diseases. And very often we work together. So here we really have something very unique to test in patients with very serious diseases like systemic sclerosis and myocarditis. But we also see effects in animal models for psoriasis and atherosclerosis and several other very difficult diseases. So the status right now in the CAN10 program is that we've done everything to be in phase 1. The phase 1 is ongoing in Germany right now. It's divided into 2 parts. So the first part is that we need to do increasing studies of single doses, ongoing according to plan. And once we reached a little bit longer, we will communicate the data. And the second part, which is important, is that it will be done in psoriasis patients. And then we will also communicate efficacy and biomarkers. So milestones, we have lots of interesting milestones in pancreatic cancer with new trials starting 24, data 25. We have lung cancer data to be communicated. The trial stopped earlier this year, and we should be able to present more. Triple-negative breast cancer, similar story. The randomized trial is ongoing. CAN10, phase 1 ongoing. And we also have several other studies which we have not communicated results from. So from an investment perspective, we will have a very exciting late 23 and 24. And by that, I'd like to thank you for your attention. I have this summary slide here to inspire everyone to ask questions. So thank you.
Thank you. And I think you have inspired people because we have a couple of questions here. First question is if you could summarize a little bit the difference between CAN04 and CAN10.
Yes. So CAN04 is designed to kill cancer cells. So it's finding IL-1RAP, and then the tail of this antibody is designed to attract immune cells to get into the tumor and kill IL-1RAP containing cells. And it's also blocking interleukin-1 signaling. CAN10 have not this feature of attracting immune cells. It's only blocking IL-1, IL-33, and IL-36.
Are you looking at further possible indications, or you're happy with the ones that you have? I guess that's a question for both the candidates.
I think there is an opportunity in lots of indications, much more than we're doing. But I think we reached a stage now where we see signals, and we really need to focus on delivering results in those diseases instead of going everywhere.
How is the work going to find biomarkers for the patients with the best outcomes?
So it's an ongoing research, and I guess what you saw in pancreatic cancer in pancreatic cancer, it's relatively easy task in front of us. We have patients with pancreatic cancer, they are always diagnosed through a biopsy. So we will have a biopsy from these patients and can measure IL-1RAP, which is one obviously very strong biomarker. But we're also trying to see if we can adapt this to let's say, more blood tests in both pancreatic cancer as well as lung cancer and triple-negative breast cancer. So we're making progress, but obviously it's very ambitious projects as well.
So, yeah, with promising results in 3 cancer forms and big potential, do you have interest from potential partners yet? Are you in discussions?
So what I can say is that my job is to make sure that we have contacts with all potential partners, and we do have potential partners who are definitely following us. And then I think if we talk from a more general perspective, I think the sweet spot for partnership is on the back of randomized data. I think that's where you can really, let's say, get the premium deals, if you call it like that. But everything is possible ahead of that. So I cannot comment exactly on where we are in the various partnering discussions.
There's also a question about competitors. Like, what are your main competitors, and how would you position yourself on the competitive landscape?
So, if we start with pancreatic cancer, I think from a more patient perspective, the competition is very limited. It's actually very sad. If you look at non-small cell lung cancer, the competition is immense. It's hundreds of different trials and compounds out there. And I think just approved drugs, it's 20 or 30 approved drugs in lung cancer. So there's no way you can try to think that you can get all patients. You need to be very clever and find your niche if you want to be competitive. And that's what we're trying to do with biomarkers, and have something which is superior to everything else.
Yeah, so these are obviously huge markets, like you say. How big a size of the market are you aiming for?
That's a great question. I think there are drugs that are even targeting, let's say, 1% of the lung cancer market that are doing fantastically well. So I don't think you have to be afraid. If you just have a good test of finding 1% of the patients and know that you're getting a superior result, that could be a tremendous market.
Thank you so much, Jan.
Thank you.
And with that, we welcome up AcouSort and the company's commercial director, Agnes Michanek. Welcome, Agnes.
Thank you. It's really a pleasure to be here with as many people in the audience to be able to present AcouSort. For those of you who don't know AcouSort, we are all about sample preparation. AcouSort has a really unique technology that enables automation and integration of all kinds of sample preparation you need for diagnostic and cell therapeutics. The company is a little bit more than 10 years old. We've been an active company since 2017, where we were listed at the Spotlight stock market. Today we are about 20 people working in the company. The technology is invented here in Lund by Professor Thomas Laurel. It's called acoustofluidics, and it is a combination of microfluidics and ultrasound that can be used to manipulate or move particles in liquids. We see that there is a huge need for improvement in sample preparation. The healthcare system today is having big challenges in how to address some of the most severe disease groups. The cardiovascular diseases, the infectious diseases, and cancers. To be able to help patients going through these diseases, you need to have early diagnostics and you need to have accurate diagnostics. And you also need to be able to give patients that are getting these diseases the right therapeutics. And AcouSort is able to provide that solution with our solutions for integrated and automated sample preparation. The markets we are directed towards is thus the diagnostic testing market, mainly for point-of-care diagnostic, where we see that out of the blood samples collected, 75% would need to have a blood plasma separation, which is something that we can provide for our partnering companies. In the cell therapy space, each cell therapy treatment is really, really expensive, which limits the amount of patients that actually get access to this advanced treatment. By making the sample preparation here more simplistic, we can spread out the cell therapeutics and enable more patients to gain benefits from the treatments. Today, most of the work that is done when it comes to diagnostic testing and when it comes to cell therapy preparations are done in the lab. Today's picks and shovels, there are the pipettes, the centrifuges, and the microscopes, and the people operating these instruments. To be able to take this to the next step, we need new picks and shovels, and that is what AcouSort can provide. So instead of having tedious labor work in the lab, we can help companies with integrating the sample preparation. And that is something that is needed to be able to discover new biomarkers, but it's also something that is needed to be able to expand new therapeutics. So sample preparation and handling, that is what we do. And we can facilitate the development of the next generation diagnostics and the next generation of therapeutics by enabling this integration. So as an example, if you look at the CAR T cell therapies, which is an area where we have been working a lot the last years, preparing a CAR T cell therapy is a complicated process. If you look at the image up on the screen, in between each of these steps, there is sample preparation. And in many cases today, that is done manual and with a centrifuge. This means that the process is tedious to perform, and it's expensive, and it has a high risk of contaminating this precious therapeutic sample on the way from the patient during the training and back to the patient. Of course, we are not the only ones that have seen that this is a challenge. Almost all the big players in the life science field working with development of therapeutic solutions have addressed this, and they are looking at ways to enclose and to make this a streamlined and safe process, and of course to decrease the cost of making the process. In the diagnostic field, what is the main challenge there is the time for making a diagnosis. So from the time the patient is coming into the doctor's office, giving the blood, until they get the feedback, the results from the testing. The reason for that is that the majority of the samples need some kind of sample handling or some kind of sample processing to be able to be analyzed correctly. Today that occurs on advanced labs, hospital labs, which means that by the time you get your results and can start your treatment or go on to further investigations, it can take up to 2 weeks. If you could use AcouSort's technology and build a diagnostic device around that, you could have all these sample preparation integrated. So when you go to your doctor, you give your blood work, you could actually get your results back while you are still there. So decreasing these 2 weeks down to 30 minutes. Being able to get your treatment started directly, or being able to get on with your further investigations without a waiting time. So what is AcouSort's technology really about? It is about sample preparation. I've said that maybe 100 times now. It's a combination of microfluidics and ultrasound, and that enables us to move particles in a liquid flow. The technology is really, really versatile. As in the example here, we are making blood plasma by pushing all the cells of the sample into the center of the channel, so we can pull out the cells in the center and the pure plasma at the side. We can also use the technology to isolate different types of cells or to wash cells, as we do in the cell therapy space. Or we can use it to select out and collect nanoparticles that contains important biomarker information that can be used to create novel diagnostics. So the business strategy that AcouSort has is to use our acoustofluidic technology and build OEM modules around it. And OEM modules are small units that can be incorporated into our partners' devices. So we provide them with an opportunity to integrate sample preparation. So today AcouSort have a few different types of OEM devices that are ready to integrate. When they are integrated into a partnering device, they are used as consumables or semi-consumables, meaning that they will start generating a recurring revenue when the system is on the market. Apart from that, we provide our partners with feasibility studies where we optimize our technology for their needs. We also have the OEM core technology built into 2 benchtop research systems that we provide to academic researchers and to companies' R&D. The OEM portfolio we have today is 3 different types of product. We have the AcouPlasma Optical, which is a small OEM component that is directed towards diagnostic devices, where you open up a small window of blood plasma in a blood sample. We have the AcouWash OEM modules that are done for blood plasma separations, cell separations, and cell therapy cell wash applications. And we have the AcouTrap OEM modules that are designed for automating sample processing for example, flow cytometry, where you need some smaller amounts of cells, or for processing and automating extracellular vesicle work. The benchtop research systems, the AcouWash and the AcouTrap, are built around the OEM modules. So we use them, as I said before, a lot in collaborations with partnering R&D departments, where they use the instrument to either do assay development or to use them to look at integration workflows. We also sell them to different universities, where we expand the portfolio of applications you can use with the systems. Some highlights from the strategic collaborations, which is something that we work a lot with from this year, is that we have an American partner that just recently got a FDA 510(k) approval for a device with our acoustic-fluidic technology inside. This is a huge milestone. This is the first time, as we know, that we have an acoustic-fluidic technology in and cleared the device for diagnostics. We also have several feasibility discussions ongoing, mainly in the space of cell therapy, but we also have a lot of discussions around automated sample preparation for flow cytometry and for sort of, lab automation, especially around the acoustic trapping module. But the biggest project we have been working on internally this year has been around cell therapy, where we had the big feasibility project looking at how cell therapy sample processing can be built in and integrated. And now in October, we had the delivery of a fully functional prototype device that the company will use in their product development to see how you can integrate our technology together with their system. And just to give you an idea about the potential of each possible OEM deal that AcouSort can create around the two different technology areas, we see that with the recurring revenue from sales of the OEM modules, even one deal can bring in a significant amount of revenue to AcouSort. So for the diagnostic field, it's about $5 to $10 million USD per deal, and for the cell therapeutic space, it would be around $10 to $60 million USD per deal. So to wrap up, I think that AcouSort is headed on a really, really exciting journey. We have a validated technology that is now in a commercial expansion phase. And we see that we are addressing fast-growing markets. And with the model where we can have a recurring revenue, we see that the business model we have is really a scalable one. And with that, I would like to thank you for your attention, and I'd be happy to take questions.
Thank you, Agnes. You have quite a few questions here, but before, yes, it's very exciting. It's always nice to walk up to the stage and see lots of questions. But we are, of course, also happy to take questions from the audience. We have Kim right there with the microphone, if anyone wants to put their hand up and ask a question in person. But we'll start here. How is your sales split between academia and industry?
For the feasibility studies, of course, it's only industry. Benchtop instrument sales, it's a combination of industry and academia. And that goes a little bit from season to season, if it's more industry or more academia.
What is your main channel to the market?
Pardon?
Your main channel to reach the market?
Main challenge to reach the market, we go to a lot of directed conferences and events where we meet, now I think I'm losing my headphone here, where we meet both investors in the medtech industry, but also when we meet academic research. So we have a lot of conference activities. Apart from that, we go to smaller meetings, where we reach out, and we try to reach out to people that are working in the space. And then I have to say that one of our founders, Professor Thomas Laurel, is a very active lecturer, and he is contributing to spreading the word about AcouSort that is really, really important.
So you go out and you meet people and...
Yes, yeah. That's the main source where we meet our customers, is by going out and meeting them at conferences, and then we start interactions there.
And we have a question here about the first feasibility project in cell therapy. When do you think AcouSort can kick off your first feasibility project in cell therapy?
We've had already a feasibility project ongoing since last year in the cell therapy space. So we had 3 different studies with this partner, and the last, or the finalization of it, is the delivery of the prototype device now. So I hope that we will be able to share a continuation on that project within the coming months.
This person has also asked how far away you are from commercial launch, but I suppose that might be a bit difficult to answer.
That is very difficult to answer for us, since we are not planning to be the ones that are launching the device. So we usually work with our partners until they go into sort of a clinical evaluation or clinical trial phase. After that, then our component is integrated into their systems, and it's to some extent out of our control to plan the launch process for our for the products that are including our components.
We have a question about competition, and it's if there is any competition outside filtration and centrifuge.
For the blood diagnostics, there is not really any competition outside of the centrifugation and filtration. I have been working even before AcouSort with trying to find new ways of preparing blood samples in a better way, and this was what I found, and that was why I was so excited by joining AcouSort a few years back, and why I'm still so excited in being here.
And I think as a final question then, if we could look ahead a bit, what can we expect from AcouSort in 2024, just to summarize?
For 2024, I think we have a lot of exciting ongoing discussions about feasibility studies, both within the cell therapy space, but also within the sort of, research diagnostic or research instrumentation field when it comes to flow cytometry. And of course, I mean, with the 510(k) approval, we are looking forward to hearing more news from our partner in US about that.
Right. Thank you so much, Agnes.
Thank you.
And with that, we will welcome up the last full-length presentation of the first session, and that's WntResearch. And the company will be presented by Chairman of the Board, Christer Nordstedt. Welcome, Christer.
Thank you, and it's great to be here in beautiful Lund. Well, it's my pleasure to tell you a little more about WntResearch and what's going on in the company at the moment. I would like to start by telling you a little about what I'm going to talk about. And what I will discuss today is our drug candidate, FOXY5, which is, to the best of our knowledge, a unique molecule, meaning that we don't really have any competition in this mechanism. And the phase 2 program that we have now for colon cancer. This is a molecule with the potential of actually preventing the spread of cancer, and I also think it's fair to say that it has potential to do some more things that I'll talk about more later on. Anyway, there's a huge unmet medical need in stage 3 colon cancer, and the commercial opportunity is, of course, very substantial. Now, the important thing here is that last summer we performed an ad hoc analysis because of some signs that our co-workers and clinicians out there had made in these patients. And it seemed like we actually saw something in this study, meaning that patients receiving active substance were downstage compared to the control. Moreover, it seems like FOXY5 has a very, very favorable side effect profile, which of course is very, very important. And what we've done now is that we have revised the study design, and primarily the dosing strategy in colon cancers, hoping that by doing that we could enhance patient outcomes. Also, and what is very important, is that we have lots of scientific evidence that this mechanism actually could be of value also in other cancer form, meaning that we have the potential here for what we could call a pipeline in a molecule. Anyway, I don't think this is news to the audience here, but we're all aware of that colon cancer is a huge medical problem out there. It's one of the 4 most common cancers actually in the world. And although early stage colon cancer usually has a very, very favorable outcome or survival, this drops quite drastically if metastases are formed. You can see here that when the tumor is in the regional state, 5-year survival is over 70%, and that drops to 14% if it has reached the metastatic stage. So if that is something that we could prevent, it would be a great benefit to patients. Well, here you see some numbers. The global colon cancer incidence is estimated to be north of 1 million per year. And of those, some 140,000 are in stage 3. And the potential sales for something that actually provides what I call clinically meaningful differentiation to these patients would be north of, or I would say way north of, $500 million per year in peak sales. Now, this is the most important slide that I'm going to show you today, and the one that I'm going to spend the most time on. And this is the observations we made in the ad hoc analysis that we made last summer. What we did was that the treatment with the substance, it starts at the stage when the patient has been diagnosed with the disease, which is done by CT or imaging. And then what the clinicians are looking at is, well, the tumor itself, the mother tumor, so to say, the primary tumor. And they also look at how it has spread in the local lymph nodes or more distally. And what we saw then after the patients had undergone surgery, which is like 2 to 3 weeks after this initial staging, was that the patients that had received active compound actually had a lower stage than those than the controlled patients. And this was analysis performed. We had, I think, about 110 patients, so 55 patients in each group. So it's a significant number of patients. And this, of course, made us very, very excited. And it made us rethink a little how we wanted to do the study. And we had to reconsider if the initial design of the phase 2 was optimal. And we came to the conclusion that it was not. As I pointed out early in the presentation, this molecule seems to have a very, very beneficial side effect profile. So we thought that perhaps we should increase the dose. And in the amendment to this phase 2 study, we're increasing the dose. And this first now, the first cohort, we had the first patient in last week. And we are very optimistic that this is something that will run on time, so to say. You know, making clinical trials meet timelines is, as you all know, very, very challenging. And then when we finished the first cohort, we switched to cohort 2 with an even higher dose. And then the idea is that we use that information from these first 2 cohorts to select a dose for step 2 in this amended study. And what we're going to do is to analyze 80 patients. So, so far, the study, the amended study, has gone quite well. As you see here, first patient have just been included, and we hope to get results during 2025. As I pointed out, we believe that we have the potential here for a pipeline in a molecule. As you can see here, there are some examples of different cancer forms where Wnt5A, which is actually the sort of the mechanism here, I won't dwell too long on that, but where it has been implicated. And as you can see, there are numerous of very, very prevalent or common cancer forms where this molecule could have a benefit. Now, you can always ask the question, did we choose the right indication when we picked colon cancer? So far, it seems like it, because it's in colon cancer that we have seen these first signals here. So we're quite optimistic about that. Now, patent is, of course, very, very important in this case. This molecule has been around for some time, but with new patents for the staple compositions, we believe that we have patent protection until 2041, and that is, of course, very, very important for us. Now, we're pleased to say that we have a talented and very experienced team running the show here, and I'll show you some of the members here. We also have a very diverse and experienced board of directors, and oops, oops, with Tommy Andersson here, who happens to be professor of medicine in the beautiful city of Lund. He's the brain behind this, and he's been with the board ever since the inception of the company. And then on top of that, we have several key opinion leaders in our as scientific advisors too, so we are very excited about. And with that, I would like to end. Thank you.
Thank you, Christer. We've had a couple of questions here about the study. So you're trying to find the right dose for the phase 2 study.
Yep.
How high can you go? And do you think the effect will get better with higher doses?
Well, I don't know. We have the pleasure of having our CEO here as well.
Exactly. We
higher
have
is
Pernilla
usually
here.
better.
Welcome, Pernilla.
Yeah, no, based on the preclinical studies and the clinical studies we have done, we increase the dose, and typically what you do is you double and you double and you double again, and this is the steps that we have outlined in the protocol, but we have not revealed the doses, but it has been approved. And the steps that one can take is defined in the protocol, and there's not going to be a long run how you evaluate. So there is a committee that will evaluate, and then we can immediately go into the next cohort, and then we can immediately also go into the selected dose later on. So everything of that is approved by the authorities.
And as you're here, Pernilla, I think we have to address the fact that you have actually resigned as the CEO. Why?
Well, I could answer that question. It's not unusual that people change jobs. I mean, it happens all the time. I've done it myself on numerous occasions. So nothing strange with that. And we're in the process now of finding a new and very capable replacement for Pernilla, even though we will miss her dearly. So we hope to have someone on board relatively soon. But until then, Pernilla will stay until a new one arrives. But as I pointed out, we have a very good, strong, and talented team under Pernilla, who is highly capable of running the show.
What are you looking for in a new CEO?
What we're looking for? Oh my God, we're looking for everything. Now, what we're looking for is someone, a strong leader with competence and experience in this field, with the ability to run clinical trials, and also very, very importantly, being able to establish collaborations. Because we realize, of course, we're a small company and we can't take this all the way. So it's a number one priority for us to establish collaborations with pharmas out there. And this is something we're working on very, very hard now, and we have lots of ongoing discussions. So it's a very exciting stage we're in at the moment.
Returning to the more clinical side of things, you recently announced a patent on a new salt formulation of Foxy 5. Why have this formulation been developed?
I think it's important to always continue with your formulation, with your substance. It's a long development. So ensuring that we have a long patent protection from the substance. So when the substance patent expire, then you have to build a use patent around that, like a patent thicket. So try to find smart ways of prolong. That's actually the reason behind it, and I think it's very, very good to have 2041 now.
So can we expect more formulations and more patents to further strengthen the protection?
Well, we need to build on this new one now, and then that's going to be the foundation moving forward.
And we have some questions on finances, of course. That the study is estimated to end in 2025, and according to the question poster here, the funding seems to last you up to mid-2024. So what are your plans in terms of financing the company?
Oh, well, that's always a challenge, of course, and especially for a molecule at this stage. I think that that's something we need to come back to, but financing is challenging. But on the other hand, you know, we're in a situation here where we're working with an entirely new mechanism, and to the best of our knowledge, we don't have any competition. And thirdly, and perhaps most importantly, we have some very promising sign that this actually could bring benefit to patients out there. And these three things are usually very good combinations. So I'm cautiously optimistic.
Well, that's a nice note to end you on. Thank you so much, Christo and Pernilla. Thank you.
Thank you.
And we will end this session with some short pitches. These are pitches from private companies. They have 3 minutes and just this one slide to present their business case. And first out is Cytely. I hope I pronounced that correctly. Otherwise, CEO Philip Nordenfelt will no doubt correct me. Welcome, Philip.
Thank you. Are you taking the time?
Linus is taking the time.
Okay, perfect. Okay. Good morning, everyone. I'm Philip Nordenfelt, CEO and founder of Cytely. And I'd like you to imagine for a moment a world where groundbreaking life science discoveries are 10 times more accurate and happen 10 to 100 times faster. So that is actually not science fiction. That is what we are turning into a reality today with Cytely. The world of microscopy-based research is plagued with time-consuming and manual processes and a lack of data analytics. So researchers often, they spend more time fiddling with microscopes and sifting through data than making actual discoveries. So that's where Cytely comes in. Our software as a service platform automates the process of microscopy-based research, all the way from image capture to data analysis. Our software steps in and controls the microscope based on what is in the sample, as well as what the researcher is actually looking for. We are a fully software-oriented platform, hardware agnostic, globally available, and we see no apparent regulatory hurdles in our path. So simply speaking, we enable researchers to do better research faster. We've already validated our solution with real-world applications and through our peer-reviewed science. And we got 2.5 million in funding from Wallenberg Launchpad through the Wallenberg Foundation. LU Innovation, Stiftelsen för Strategisk Forskning, and Almi are some of the supporters on our list, and we are part of the SMILE incubator program, of course. Shout out to my SMILE friends in here. Our business model is simple but scalable. So, as I mentioned, we're a software as a service oriented platform, and we offer tiered subscription plans based on each individual research need. So whether you're an individual scientist or a large research group, we've got you covered. The market for life science software is a multi-billion-dollar market, and we do not intend to just be another player. We're aiming to revolutionize this space. So the team consists of expertise within cell biology, software engineering, microscopy, and entrepreneurship, and we have a great mix of talents to really drive this venture to a success. We're seeking a 2 million Swedish crown pre-seed investment, and this is to expand on the product, the team, and grow our customer base. So join us today to set forth on this revolution in microscopy-based research and help us manifest our vision of democratizing microscopy.
Thank you, Philip. Thank you. Next up is TillaCare and CEO Mikael Kuhn. Welcome, Mikael.
Thank you for letting me present. So I'm Mikael Kuhn, I'm CEO of TillaCare, and I want to talk to you about severe urinary incontinence. It's affecting millions of people globally, and it's a market worth about $20 billion in the world. However, severe urinary incontinence is 5 to 6 times more common in women than in men. With almost 20 million women in the US and growing, it's a really big problem. For the women, there is no solution. For men, there is a specific solution apart from the traditional diapers with all its problems with sustainability and wounds and the traditional invasive catheters causing infections. We at TillaCare has changed that. We created the female condom catheter. It's a sustainable solution designed specifically for women. It's changed once daily, it's soft, and it's made out of silicone. We found that the smooth surface of the silicone fits and creates an adhesion around the urethra of the female, in the female anatomy, and seals from the environment. You can think about 2 smooth surfaces of glass with some liquid in between. You can move them sideways, but you cannot separate them. We can just peel the soft silicone out of there without hurting the patient. It's also, because it insulates from the surrounding, it's also uniquely suited to take non-contaminated urine sample from women, which is a huge problem for incontinent women to really get a non-contaminated sample. The UriCap was introduced in the US market 2022, and we have regular sales expecting this year to around $160,000 with our first and second distributors. Our team reached a lot of milestones, even though there was COVID. We have regulatory approvals in the FDA, CE, Brazil, Australia, and in the UK. US and EU patents are in place and some other places too. And we are the only reimbursed external urinary catheter in the US with Medicare reimbursement at $12 a day. And it's changed daily. The device is changed daily. We also, from February, have reimbursement in the UK through NHS, and the distributor there is already started to market. Manufacturing is in place with a very attractive gross margin, and we already started enrolling other distributors in Australia and more distributors in the US, targeting the large home market in the US, and in other markets it might be home and hospital markets. Our key drivers are, of course, that we're the only reimbursed external female catheter in the market. And we're also helping the distributors to reach into the female sector because they're only selling to men today, so they can increase their business. So it's a very interesting business proposition for them. We are looking for Series A investment. We hope to reach cash flow positive by next year, and with the revenue stream model after our adoption of our first distributors, we think we can have a $12 million revenue stream 2026 with about just 16,000 patients, very few patients on a multi-million-dollar patient market. And I think that is a very attractive proposition to the investors. Thank you.
Thank you. And our final short pitch will come from Redley Diagnostics and CEO Lisa Lööf. Welcome, Lisa.
Thank you. And thank you for letting me come here. Hello, my name is Lisa Löv and I'm the CEO and founder of Redley Diagnostics. Imagine that you get sick, really sick, and you can't breathe. It gets harder by every time you try, and in the end you just can't get any air through. You rush to the hospital, and once you're there, you want to find out what's going on now. Acute respiratory symptoms require actions. And today, diagnostics is done with PCR, and it's a super good method, but it requires heavily instruments and trained personnel, and it also takes time. We at Redley want to offer immediate care with our respiratory panel. We are 100% instrument-free. This is what we're making. It's a lab by your fingertips. We have near-patient testing, simple yes or no answer to an ongoing infection of any of the 7 pathogens we can detect with this panel. This is all you need. It takes minutes, not hours, not days, and our technology, we have 2 patents filed, is as sensitive as the PCR. We went out and asked clinicians what they thought about us, and they see that we have a clear position in the emergency room and at the elderly care. They want us to be there. They can't wait for us to come there. So our plan is to have a CE IVD marked test, produce this in our in-house production line in Uppsala, Sweden, and then find a partner for distribution and sales. And today we are at this phase, we are at prototype phase. We will have clinical feasibility within the year of all the 7 pathogens included in the panel and a functional prototype. We will have pilot production within a year, followed by clinical trials and CE certification by the end of 2026. We'll be on the market by 2027. But we already have lined up customers waiting for in the research field, and they are excited to test this after the pilot production. So we have a capital need of 50 million SEK for the coming 3 years, and we aim to take that in between private investments and also soft money. And if you want to hear more about this amazing journey that we're on, please come and talk to me. Thank you.
Thank you.